From Microbe TV, this is Immune, episode number 56, recorded on May 25th, 2022. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back.
Hello again, Cindy,
Hello.
Having recently been here.
I did. I came down to the incubator. Yeah, that was really fun.
It was a fun time.
Yeah.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see you all. It was great to see both of you in person recently, and now great to see you back virtually.
Yes, Brianne was also at the incubator launch. And we have a very special guest today, who I understand Cindy had to pull in all her favors, to get to join us. He is the Chief of Experimental Immunology Branch at the NIH. Al Singer, welcome to Immune.
Thank you very much, Vincent. I'm glad to be here.
So, Cindy, you have no more. He doesn't know you anything else. This is the last time.
I'm out of favors.
Ah, too bad.
I'll have to come down and take him out for coffee. Then maybe I'll get another favor someday.
So last week, Cindy said, "Oh my gosh, this paper is burning up the internet. Maybe I could get Al on." And so here we go. It's great. So we're going to talk about your wonderful flip-flop paper. But first, please briefly tell us a little synopsis of your history. Starting from, I'd like to know where you're from. You're not from Brooklyn, are you?
I started off in Brooklyn.
Look at
as
that.
a young.
You could tell the accent.
But Brooklyn, but Brooklyn at that time was always a place people left. I'm stunned that I have a son who's decided that Brooklyn is a place to go to. So he's there now. So
what were you doing in Brooklyn? You were not born there, right? I
was not born in Brooklyn, but we emigrated from Europe then, and we lived in Brooklyn. I lived in Brooklyn for 6 years, and then moved to a small town in upstate New York.
Where in upstate New York?
There's a place called Port Jervis, New York.
Okay. Oh, yeah. I am from a small town in upstate New York, so that's why I asked.
Probably much more upstate. Where are you from?
Camden,
New York? Where is that?
It's near Utica and Rome.
Oh, so you really are upstate. This one is not nearly that far. Yeah.
Where did you go to college, Al?
I went to MIT.
Wow.
And then I was at Columbia in medical school.
Right.
Mm-hmm. And this was in what decade were you talking about here?
I mean, what century? Yeah, no. So I graduated medical school. I graduated college in 1968 and then 1972 in medical school.
Okay.
Yeah.
Yeah, I was not there yet. I didn't get there till 10 years later. So tell me one person you remember from medical school. Do you remember Harold New?
Of course I remember Harold New.
Yeah, I thought so.
Do you remember? You remember Harold New?
Obviously. Yeah. He was there, and when I lectured medical students, he would sit in the front row and tell me when I said things that were wrong. He never hesitated, right?
Yeah, he was always an impressive fellow, actually.
He is.
Medical school.
So do you remember any immunologists from Columbia days?
I do. There was a fellow named Vince Butler. I don't know if he was still around when you got there.
No.
He had made antibodies to digoxin and digitonin.
Okay.
And which at that time was a big deal because it allowed us to measure. Yeah. So there were a number of immunologists. Columbia always had thymic aplasia. It was very interested in antibodies and proteins that circulated, and very uninterested in where they came from. So it was an odd mix.
So I remember two immunologists, Elvin Cabot,
of course,
and Ben Pernis.
Yes. So Ben Pernis came after I was already there, and Elvin Cabot. So Elvin Cabot was the one. So he was the one who really provided the focus at Columbia on antibodies.
Yeah.
And so So it was very weird. As a medical student, we could do quantitative immunoprecipitating curves, but we had no idea what a cell was that actually produced any of this stuff. It was odd. It was very odd.
What did you do after Columbia?
So I did an internship and residency at Presbyterian Hospital, and then came down to the NIH, and I've somehow gotten stuck here for the rest of my life. I walked into the lab.
Not a bad place to be stuck.
No, I quite agree. I was always frustrated in medical school because I never really felt like I understood what caused disease. And when I got here and started studying immunology, there was a satisfaction that I just never had before. So that's what I've been doing since, actually.
Such a contrast between your days at Columbia and now the amazing technology that you have, and we're going to talk about today in your paper, right?
Well, things have changed. Things have changed. I don't know that the problems have changed as much, but our ability to study them and to address them have certainly changed.
So I actually have one question, Al. Do you remember, because this is one of my favorite memories of being there. Do you remember you used to have a picture of a mouse outside of the lab, and you would ask people, what's missing? Do you remember this?
I don't, actually.
And the mouse was lacking a thymus. and so basically it was like a trick, right? You'd go and you'd big people in the hallway like, what's wrong with this mouse? But I had talked to people in your lab and they had told me what it was. And so we were standing in the hallway and you said, what's missing on this mouse? And I said, that's weird. It doesn't have a thymus. And you were like, oh. And I was like, pulled one over on you.
Did you win the prize?
He probably doesn't remember that. But that was really funny. Yeah,
Did you tell by looking at the mouse that it didn't have a thymus?
Well, yeah, it was like splayed open, right? And it was like what the different organs of a mouse.
That was an actual mouse, not a schematic.
Yeah. But yeah, so that was pretty funny. The other thing I remember is Al would interrupt everybody on the very first slide, no matter who it was, when they were giving a presentation. Do you still do that?
I never realized I ever did it, but I guess I did. I really do on the very first slide, huh?
Yeah.
Okay.
Because you're just so interested and engaged, you know?
Yeah. No, he was a fantastic branch leader, and NIH was a great place to do a postdoc. Learned a lot. Yeah.
I think that should get you more points, Cindy.
Yeah, definitely. Cindy,
I earned back another favor.
Yeah, I think you get to ask the inaugural question about this kicking off the paper, since you were the one who got the thymus right on the mouse.
So the paper we're going to talk about today is called Reversal of the T-Cell Immune System Reveals the Molecular Basis for T-Cell Lineage Fate Determination in the Thymus. And it's, of course, from Al's lab. And there are a couple of names I recognize on there that are there from when I was there. But so before we go into the paper, I actually posted on Twitter and I asked people if they had questions for you. And a common question was like, how did you ever get an idea to do this? So we're going to talk about exactly what you did. But first of all, this would not have been possible without some of the more advanced genomic technologies, because what you in fact did was take the CD4 gene and pull it out and put it in the CD8 locus, and take the CD8 gene and pull it out and put it in the CD4 locus, and make flip-flop mice. So what prompted you to do this?
So actually, it actually made sense to do. I'll try to explain that. What we actually did is we took the protein that the CD4 gene and we dropped it into the CD8 gene.
Yep.
So the CD4 protein was made by the CD8 gene, and we took the CD8 proteins, in that case both alpha and beta, and dropped them into the CD4 gene locus. And the reason we did is that we came up with a model years ago. Actually, we published a paper in 2000. The first author was a fellow named Enrico Bruniera, who came from Switzerland. And what Enrico had discovered was that when you took double positive thymocytes, these are the immature cells that give rise to the mature T cells that eventually become the mature T cells that populate the mouse. That if you signal the double positive thymocyte, it's an immature cell, it doesn't know what it's going to be. It expresses both CD4 and CD8. It has lots of potential, but no function yet. And the question was, how do you get this cell to how do you signal the cell so that it becomes a mature T cell? And how do you particularly signal it so that it becomes a helper cell or a cytotoxic cell or a CD4 cell versus a CD8 cell? So there were a lot of mysteries then. And so and we knew very little about it. So virtually any experiment you did, you always learned something. You didn't always understand what you learned, but you learned something. And what Enrico found was when we signaled the cell, no matter how we signaled this double positive thymocyte, the same thing happened, which is that the signal turned off the CD8 gene and it turned up the CD4 gene. So the CD8 gene got turned off, the CD4 gene got turned up. So we tried to understand what did that mean? Mm-hmm. And there are lots of models of information theory about how do you convert that kind of information into rules that the cell can now live by to decide what to do. So it was clear that the cell doesn't know whether it's being signaled by CD4 or CD8, but it knows it's being signaled because when it's signaled, it suddenly loses the activity of the CD8 gene and it upregulates the CD4 gene. And so the model that we came up with was a model that we called the kinetic signaling model. And what we said was that basically the lineage choices that the cell could make were directly a result of the cell interpreting the fact that it had now been signaled. So it loses the CD8 gene, it upregulates the CD4 gene. And so if you have a receptor that is now, for example, seeing a class 1 molecule, it sees the class 1 molecule in association with the CD8 coreceptor, because the CD8 coreceptor sees class 1. So if you lose the CD8 gene, you lose the CD8 protein, and suddenly that signal disappears. So the cell knows or interprets the loss of a signal as being dependent on the fact that it had been signaled now by a CD8 class 1 signal. And the way the cell knows that it's been signaled by a CD4 class 2 signal is because the signal is not lost, it persists. Okay, does that make sense at all?
Yeah.
That's a really helpful explanation.
Yeah. Makes a lot more sense.
Yeah. So the cell doesn't have to interpret, doesn't have to figure out a way. Do I know this is class 1 or class 2? Do I know this is from the CD8 coreceptor or the CD4 coreceptor? It simply has to know that the signal that was there is no longer there, or that the signal that was there is even stronger than it was before. So the kinetic signaling model says that if a signal is gone, it's a CD8 dependent signal, and the cell realizes, aha, I turned off CD8, but I need CD8. And it actually goes through these maneuvers to turn CD8 back on again and to turn CD4 back on, turn CD4 off. Or alternatively, if the signal persists, it says, aha, I actually don't need the CD8. I had turned it off. I don't need it. And it leaves it off and the cell becomes a CD4 T cell. And then we learned from the transcription factor people that, in fact, the lineage, so for example, the persistent signal that you got from the CD4 class 2 signal, that would turn on TH-POC, which drives the cell to become a helper cell. And if you lose the signal, that turning on the CD8 gene again is done by RUNX3, the RUNX3D transcription factor. That is the lineage fate factor for the CD8 cytotoxic lineage. So the kinetic signaling model is how all this works. We've been convinced of this for a long time. Mm-hmm. It's been very difficult to convince my colleagues about this.
Well, because there's another model, right?
Well, there have been quite a number of models. You know, if you teach immunology, you probably have taught the instruction model, and you had the stochastic selection model. Mm-hmm. You know,
Kubi still has a nice figure of 3 different models.
Yeah, so the instruction model was that somehow the signal that you got from the TCR and co-receptor told the cell to turn off the CD4 or the CD8 gene. Yeah. And it's a very attractive model. The only problem is that if you try to find something different about the signal you're getting from the CD4 or the CD8 gene locus for this TCR signal, you don't find any differences. There's nothing you can find. So how come we can't see a difference and the cell understands the difference? It doesn't make sense. The stochastic selection model was a popular model too, where it argued that the cell has no clue what the nature of the signal was, so it randomly turns off one co-receptor or the other and sees what happens. So if you turn off the co-receptor that you need, the cell loses the signal and dies. The problem with that is that you lose half of your double positive thymocytes. So having gone to a tremendous amount of trouble to rearrange T-cell receptors that actually would now bind to a ligand, to lose half of the ones that were functional by simply inadvertently turning off the wrong co-receptor would be awfully wasteful. So it was simple, but again, when you looked, you couldn't find evidence in favor of it. The kinetic signaling model tells you don't need instruction. You know, the way this is really working is whether the signal, so built into the system is that the TCR signal turns off CD8 selectively. So that signal is always lost, the CD4 signal is always on, and the cell then goes from there. It knows what the signal is because of whether the signal is persisting or not. So what controls whether the gene is on or off are the regulatory sequences in the CD4 and CD8 gene. So the prediction from that model is that if you switched the proteins that are made, it still shouldn't make any difference. Mm-hmm. The CD4 gene will still stay on, the CD8 gene would turn off, and the double positive thymocyte would know what to do from there, but it would now be doing it with different proteins than was there. So it really was a straightforward prediction from the original paper back in 2000. It's just that it's taken us 20 years to develop the technology to be actually change the gene products for both the CD4 and the CD8 gene.
So you fundamentally believe that you were going to make CD8 helpers and CD4 cytotoxic cells.
Yes. Mm-hmm. I mean, I'd love to say that we were just shocked by this,
but you were just did it. We were right.
So basically, the idea is the cell doesn't actually know anything about the CD4 or CD8 protein. It knows, that thing at that locus I just turned off was useful.
Correct. Exactly right. That's exactly right.
Right. And so then evolution with CD4 and CD8, the protein structure and what it was doing for the MHC interactions, that was evolving independently, probably, of the locus.
Correct.
Signaling. Right.
Correct. And yes, that's correct. That's a future paper, by the way.
Okay, I was wondering because my other question, which could have been asked at the end, was what other proteins could you put at the CD4 and CD8 locus? I mean, you had altered function because when you flip-flopped them, for example, antigen presentation wasn't sufficient. You can't do cross-presentation. But if you put other proteins there, could you still get at least the first half of the story, which was, well, you got T follicular helper cells, for example. I'm getting a little too into the paper, but So some questions I had.
Yeah, so you're right. You could put other proteins in, and but as you study this thing, and as we've studied the flip-flop mouse, you find that there's obviously there's coevolution of the genes and the protein.
Right.
So, for example, the CD4 regulatory sequences are making a very powerful co-receptor, the CD4 co-receptor protein, whereas the CD8 gene locus makes a very weak CD8 co-receptor protein. So the end result is the transcription of the CD8 locus, because it's making I think it's because, I don't know that this is the reason, but the CD8 locus, which makes a very weak co-receptor protein, makes a lot of it. So you have a very high level of CD8 transcription of a very weak co-receptor protein. The CD4 locus is you make relatively little of a very strong co-receptor protein. So which is fortunate, because because otherwise the system gets really out of kilter. If you make a tremendous amount of the CD4 protein and very little of the CD8 protein, you really have you have an imbalance. And what you're saying is, if you look in the flip-flop mouse, you see an imbalance.
Yeah.
You have you get lots of CD4 T cells, and you get very few CD8 T cells, which for us was actually a good thing, because here we had a very weak co-receptor, and it was expressed at very low levels, and yet those cells still became helpers.
Right.
And so since almost all the people who are listening to your podcast probably tuned in, if they thought about this at all, they thought, yes, strong signals make helper cells and weak signals make killer cells. And you look at this paper and something's wrong, because here you have very weak signals, even low-level weak signals making helper cells, and the opposite for the killers. Yeah. So that's the idea. Yeah.
Yes.
Sorry. I have a question about the setup of the mouse and the way that you were designing the mouse. And so you have CD4 in the CD8 alpha locus and CD8 alpha and beta in the CD4 locus. So
Correct.
I guess my sort of two questions are, what about the CD8 beta locus? Does that sort of change the relative amounts of CD4 and CD8? And what about cells that are normally non-T cells that are expressing CD8? Are they impacted? And have you looked at those?
Yeah, those are all very good questions. We chose the CD8 alpha locus because much more is known about the transcriptional regulatory elements for the alpha locus than the beta locus. And it's a very weird locus. Many of the regulatory sequences that control CD8 alpha are actually embedded in the CD8 beta gene. And over the years we've made many mice that didn't work, because when you futz with these regulatory sequences, you never really know what the consequences are going to be. And so we were worried about futzing too much with beta, because we didn't know it regulated beta. So that's why we did alpha. It's a very simple reason. We also made sure it had both alpha and beta, because we knew to be able to bind to class 1, you need to have both CD8 alpha and CD8 beta. So just very practical reasons for doing it. I don't know what would happen if we did this for beta, but it makes a good.
Yeah. Have you looked at the other CD8 positive cells, like dendritic cells? Are they normal?
They're normal. We haven't seen abnormal expression. It's a very good question. We haven't studied them in great detail. That paper has not everything that we've done so far, but it has a good part of it, a lot of it actually, what we've done. So are you surprised that that things like the dendritic cells aren't screwed up by having a different co-receptor protein made by their CD4 and CD8 gene loci?
I don't know. Are they actually CD4 positives? Did you look? Or do they stay CD8 positive? Because if the expression of CD8 in the dendritic cells is dependent on the locus, one would think that when you flipped it, maybe they would be CD4 positive.
They should be, and I don't know that we would be able to tell. I don't know that we've looked carefully.
Okay.
I can tell you that when I've given this, when I gave a seminar, and the first question I was asked was, okay, so what happens if in addition to switching CD4 and CD8, you now switched class 1 and class 2?
So are you going to do that?
Those are some big loci to mess with.
Yeah, I'm a little bit too far along to do that experiment.
Take years.
Yeah, but it's an interesting thought. But you raise good points here. I don't know whether I presume the co-receptor proteins are being made by the appropriate locus, so they would be switched in the flip-flop.
Right.
Yeah,
and we know for sure, right, for the dendritic cells, CD8 positive and dendritic cells, the only reason why the CD8 is there is so we can use the antibodies to purify them, right? Obviously.
That's a good reason. It's a very strong evolutionary reason.
Yeah. So if we want to talk about a little bit about the data in the paper, so what were some of the first things that you were interested in looking at when you made these mice? So first of all, do they have T cells, right? And do they have the appropriate numbers of T cells? And can you talk a little bit about the initial characterization of the T cells that you did?
Yeah. So we wanted to know if the thymus was, if the structure of the thymus was changed. So the thymus has a cortex and a medulla. And we wondered, is it possible that changing the CD4 and CD8 gene products change the structure of the thymus? So we looked. And the structure of the thymus, not surprisingly, was normal. It looks just like a normal thymus. Nothing changed in terms of the structure of the thymus. We looked in the periphery. We saw CD4 and CD8 T cells. They were in the normal places. And that was all normal. The CD4 T cells normally react to class 2 antigens. And these flip-flop CD4 T cells react to class 2 antigens. So they were normal in their recognition specificity. The CD8 T cells, which normally see class 1 antigens, reacted to class 1 antigens. So they were normal in their specificity as well. So everything looked normal. The T cells looked normal. They had they were in the right places. Their numbers were changed, and that was largely because the expression levels of the CD4 and CD8 co-receptors changed. But what was really striking was the thing that was different was they had a completely reversed function. That the CD4 T cells were not helper cells. They were cytotoxic cells. And the CD8 T cells, instead of being cytotoxic cells, were helper cells. And what's really interesting about the mice is that most of your listeners believe that class 1 and class 2 does very specific things. So, for example, the regulatory T cells have to see class 2 because they need a strong signal and they need to see class 2. Well, these CD4 T cells have a C class 2, but they're not regulatory. But they do have regulatory T cells. Mm-hmm. The regulatory T cells are CD8. They're class 1. And yet they express FOXP3. And they suppress. And they function just like regulatory CD4 T cells. But they don't see class 2. They see CD8. And you go through all these different things, and what you realize is that so much of what we understand is what we've seen. And we've presumed that what we've seen has to be seen. But in fact, doesn't have to be the way it is. So in the flip-flop mice, things are different. The regulatory T cells are CD8 FOXP3. The follicular helper T cells are CD8 T follicular helper cells, not CD4 T follicular helper cells. It's very weird. It's very weird. But I can tell you it takes a long time to try to figure, just to understand the mice, because it's so ingrained in our understanding of immunology that having CD4 and CD8 function reversed is such a confusion.
It really is.
It's such a confusion. I think you, Cindy, you mentioned in your email that one of your students thought this was mind blowing. It really is mind blowing, and this is the consequence of having your mind blown.
So yes, I feel like there are multiple times where I've had to kind of sit and process different parts of this paper as I've been thinking about the model. And as you were just saying something, it gave me another one of those moments. So just to check that I'm understanding correctly.
Yeah.
So with the regulatory T cells, they could still potentially have strong signal from through the TCR in terms of interacting with MHC plus peptide, but they, upon receiving that signal, upregulated CD8.
Yes.
Okay. So it's a change in the signal from CD4 or CD8. The strong signal from TCR is still a thing. It's just whether or not they put CD4 or CD8 with that strong signal.
Yes. So I hope I didn't confuse you by my comment.
I got it now.
Okay. I should stop then.
So in other words, if you have strong reaction between your T cell receptor and the MHC, you've got basically two choices. If it's too strong, you get negatively selected. But if you're kind of on the border, you become regulatory. And it doesn't really matter whether that strong signal is coming through CD4 or CD8 co-receptor. Is that
It does not matter whether it's coming through CD4 or CD8 co-receptor.
So
it's easier to get a strong signal coming through CD4,
right?
Because the CD4 co-receptor protein is a much stronger co-receptor protein.
So you're still reacting strongly with self. You are still making Treg cytokines, but you happen to have CD8 on your surface.
Right.
And you have a T cell receptor that is C in class 1.
Right. Okay.
Right. And so one of the questions then is how much is that getting triggered in the periphery? Because if you have CD4 helper cells, presumably the re-triggering isn't happening as frequently than if you have CD8 seeing MHC class 1, because that's basically everywhere, right? So if it's recognizing some sort of peptide that has low affinity or whatever, but it sees it everywhere, how does that change potentially the function of that regulatory cell compared to if it occasionally gets re-stimulated?
Yeah, that's a really good question too. And I really was curious about the answer to the question. So one of the experiments that really surprised me in this paper was we introduced the Scurfy mutation into the flip-flop mouse. So the Scurfy mutation, the animal basically doesn't make FOXP3. So in a normal mouse, you get an autoimmune disease because you no longer have functional regulatory T cells. And what's interesting about that is that if you don't have FOXP3 regulatory T cells in the normal mouse develops a very relatively rapidly fatal autoimmune death. So the animal lives for 3, maybe 4 weeks and then dies. So we wondered what happens in the flip-flop mouse, because the Tregs now are not seeing class 2, they're seeing class 1.
Right.
Do they also get autoimmune? And I was quite surprised. What we found was that the Scurfy flip-flop mouse is also autoimmune. And that mouse also died in 3 or 4 weeks. So even though there are relatively many fewer of these regulatory T cells that are CD8 and that are seeing class 1 than you have in the normal mouse, in the normal mouse, you still require those guys to be present to avoid this autoimmunity. I was surprised by that. If you look there are pictures of the lymph nodes. The lymph nodes are big and juicy. They're huge in the flip-flop mouse. Yeah, it's curious. Isn't it curious?
It's very curious.
What we thought we knew about the importance of class 1 and class 2 and CD4 and CD8, I think a lot of it is not true.
Right.
I think what it's changing is we were thinking about the quality of the signal, like what signals are happening and what molecules are interacting. But really it comes down to almost a computer, right? It's either on or off, and there's not as much distinction as one would have thought.
So yes. So I think that that's a very good and very succinct description of the kinetic signaling model. That the information is not qualitative. It's not that the cell is being told, or you have different qualitative signals that tell the cell it's class 1 or class 2 or helper to become helper, regulatory, this and that. It really is primarily whether the signal is there or it's not there. And that's in in this computer age, this should be a popular perspective. Because it's really a binary, it's really exactly that. It's a binary system.
But what makes it more complicated is, though, it's not one binary, right? We've got many, many different binaries because you've got a binary on all different signaling molecules or transcription factors. But it still comes down to on or off.
So the developmental, well, it's what happens is that timing is everything in life. And so for the double positive thymocyte, the time in life is at that moment of positive selection. Does it have a receptor that can signal the cell? If it has a receptor that can signal the double positive thymocyte, the cell survives and starts to differentiate, and it has to make a decision: what am I going to become? And so your high school senior is coming to that point. What am I going to do? So it is I think a lot of the qualitativeness comes from the timing of when these different signals occur. Mm-hmm. That's what I think. That's pure prejudice, though, I have to say. Yeah.
So you had talked about some of the functional experiments that you did, and we were talking about T regulatory cells, and that was very curious. And I think the next one in the paper is the rejection of the skin graft, and it was the same in the flip-flop mice and the wild type. And so, I guess, was that expected? And what did you think about that experiment? I think I would have hypothesized that would be expected. Some of the virus experiments, maybe not so much, but what were your thoughts on those experiments?
Yeah, good question. Well, we were hoping that the skin graft would be rejected. Mm-hmm. The skin graft rejection in the mouse is probably the strongest immune response you can elicit.
Right.
And so we thought if we had a functionally competent immune system at all, it should be able to reject skin graft, because you have the individual cells can reject skin. CD4 and CD8 T cells alone are sufficient to reject skin. If you have both of those cells, they can each reject skin. There doesn't have to be many interactions. So we expected the skin graft to be rejected.
Right.
At least we hoped it would, as an indication that the immune system that the cells were functional.
Right.
And that's what we saw, that they did reject the skin. We didn't do anything particularly insightful. I'm sure if we got into it and were more careful in our analysis of the skin graft rejections, I'm sure we'd find many deficits in the ability of these flip-flop mice to reject different kinds of skin with subtle differences. Mm-hmm. Yeah.
And coming to something that Steph is really interested in that I wondered about, I mean, I assume it's fine, is actually their survival through birth are they born at normal Mendelian ratios? Is there any sex difference? Is pregnancy normal?
I think the answer is yes to all of that. I can't say we've studied or looked at it very carefully, but we haven't had problems with the breeding or anything else of these mice. What I found interesting was something we haven't gotten to, which is, so how was it that evolution made the CD4 gene encode the CD4 co-receptor protein and the CD8 gene to encode the CD8 protein?
Right.
Because we did this experiment, and the question is, did you think Mother Nature never did this experiment? I presume Mother Nature did this experiment. She looked at the results and decided this is not a good idea. And it was not a good idea. You don't make good antibodies,
Right.
because you can't get T cells, the T follicular helper cells, even though they're perfectly functional. You can't get them to interact with antigen-specific B cells.
T cells, yeah.
Yeah, and it's not the T cells' fault. And it's really not the B cell's fault either. The B cell is just incapable of taking soluble antigen and taking it inside and presenting it on class 1.
Right.
And the helper cells in the flip-flop are only recognized class 1. So they can't interact with the B cell, so there are no antibodies. What came as a bigger surprise to us is when we looked at the antiviral responses in these flip-flop mice. You can't clear virus. You can get T cells to recognize the virus, but they don't clear the virus. The T cells that are recognizing the virus, so when you look at the CD4 T cells, these are killer cells.
Right.
They see class 2.
Right.
And they kill the antigen-presenting dendritic cells that pick up the virus.
Oops.
So,
I have questions about both of these experiments, because these experiments for me, I spent a lot of time thinking about. I guess to go back to the antibody experiment and to the B cell experiment first. So I was surprised by the data here, because I assumed that when I think about, say, making an antibody response and linked recognition, it seems as though the cell the B cell is internalizing antigen and is presenting on class 2, of course, to turn on a CD4 helper cell. But it should be presenting on class 1 as well. Other stuff, self, for example.
But only peptides of proteins that it synthesizes in the cell.
Right. And so I guess is the problem that there just aren't helper cells, class 1 helper cells. I don't know what we're calling them.
That are semi-autoreactive?
Yeah, there aren't enough semi-autoreactive class 1 helper cells to provide the help in that case.
Well, you can get, so there may be. The problem is that if you normally, so this is now antigen non-specific, right?
Right. So
because they would
I'm
not be
thinking,
seeing the same antigen that the B cells are reacting to.
Right, but that's
so you want them
we
to
see that in a hapten carrier situation anyway.
In a hapten carrier situation, you're usually looking for the anti-hapten response, right?
Yes, on the
you
B cell.
get anti-carrier
and
on the
T cells see the carrier.
The anti-hapten. Well, so that's the lore, and they do see the carrier, but of course, T cells don't know the carrier from the hapten, so they'll see that. Whatever the T cell is seeing, it's called the carrier. We call it the carrier, and the B cell is seeing the hapten. But the B cell's not making anything. So if you polyclonally, the way you get is what you want is you want to have the T cell physically contacting the B cell. Mm-hmm. And the T cell, the T follicular helper cell, is going to activate the B cell. It's going to cause it to undergo class switching. Mm-hmm. It's going to undergo somatic hypermutation. And so you get a higher affinity antibody, and you you go from an IgM to an IgG or some other class of immunoglobulins. For all that to happen, you have to have an antigen-specific T follicular helper cell to actually physically contact and we use words to explain concepts we don't understand. But we want to have that T follicular helper cell see the antigen that the B cell has taken up on the cell surface so that it can interact with it directly. And for class 1, it doesn't happen because the B cell can't take the exogenous antigen, the haptin, and put it onto the class 1 molecule. Then dendritic cell could do it.
And so that's why you get T follicular helper cells, because of the cross presentation to allow those cells to come about, to maturate, but they cannot interact with the B cell, so you're not going to have antigen-specific IgG production.
Yeah, I think what I'm realizing
Correct.
is that exactly as you said, this was the lore about the haptin and the carrier not always having to be the same antigen, and this is challenging that lore.
Right.
A bit from...
Well, I think... okay. Yeah.
Maybe I'm misunderstanding.
So pivoting a little bit, I have a question. I would have thought that these mice would have problems. And the reason, specifically in the intestine, because my understanding is that you really need good IgA production in the intestine to be able to control the microbiota. And if you can't class switch, you can't get IgA. If you can't get IgA, you can't control the microbiota. And so I would have thought that these mice should have some inherent problems, like commensal bacterial transmitting and causing nosocomial infections and causing problems. But did you see that? I mean, were they just generally healthy, or did you euthanize them or use them for these studies so early that those things might not develop, and if you aged them they might? Or
I don't know.
I think you raised some really good questions. No, I don't know that we've never seen anything. No, it's okay. We never looked. We never looked, Cindy. I think it's somebody. You raise good points. So if you think there really are something abnormal with these mice, yeah.
It's interesting because IgA deficiency is one of the most common deficiencies.
1 in 200 people, right?
And they actually are quite fine. A lot of people might not even know that they're IgA deficient, and what the experiments and the literature was suggesting is that you still have IgM that has a secretory component that can make it to the mucosal surface. And because these mice are still producing IgM independent of interacting with B cells, that you would, I'm assuming that any deficiency that you might see without IgA, T cell dependent IgA, that you would fill that in with IgM. That would be my hypothesis. Right. Be interesting to look further.
Fair enough.
Well, we'd be happy to send you the mice, and you
can
and take a look and see.
We're just giving you more work to keep you busy.
Yeah.
Well, thank you very much. But as I said, I'm pretty far along in this game.
Well, maybe one of your past postdocs who has who started their own gig, they could do this. Going back to the virus experiment, I don't know if Vincent has questions about this. You said you were surprised because there was a lack of viral clearance, partly because these cells, the cytotoxic CD4 cells, are killing antigen presenting cells because they're presenting the antigen, and those cells know the antigen and they're cytolytic. So yeah, that's intriguing. I forget what the other hypothesis was. There was kind of two.
Yeah. When I looked at these data, I was again really struck by what this said about the importance of CD4 help for CD8 cells, because what I was imagining was that the CD4 helper cell, which in this case would be the CD4 cell, is trying to help that CD8 cell. But when it tries to make helper cytokines, instead it makes perforin and kills when it was trying to help.
It's almost saying, I'm sorry, I'm trying, but these people, they made me produce the
all
wrong
I do
thing.
is kill. I can't help.
So why is it when you knock out perforin, then the virus is cleared? I mean, I understand it's now no longer killing these cells, but what is clearing it? I thought perforin was important for clearance.
Perforin is important for clearance by cytotoxic T cells, for sure.
Mm-hmm.
I don't remember it that if you get rid of perforin, you now clear the virus. I
That's
don't
what
think
you say
we ever.
here. Said you say interference with viral clearance was specifically mediated by flip-flop origin CTLs because interference did not occur. That is, virus was cleared when the origin flip-flop T cells were derived from perforin.
Yes, so that was in a very complicated system where we had a mixed chimera. We had a mouse that had normal conventional cytotoxic T cells from the B6 origin.
Oh, okay.
And then we had our interfering flip-flop guys.
Got it.
So the flip-flop guys prevented the clearance when they were allowed to be cytolytic. When we took away perforin, then they no longer interfered, and so the virus could get cleared.
But those mice had normal perforin positive CTLs, right?
Yes.
Okay.
Yes.
Got it.
So I always worry that people who read the paper will feel they know less now than they knew before they started.
No, I never knew anything so it doesn't matter.
That's not true, Vincent.
I love that though, right? Because I think it really does challenge. Well, like you said, the kinetic model has been one of the proposed models for a long time. You've been working on this for 22 years since that 2000 paper. So in a way, like you said, it's not surprising. You said that, you weren't surprised, you were expecting this, but it does really open this kind of Pandora's box of what are these cells, right? Why did the protein and the gene locus co-receptor expression, they've co-evolved, but they've evolved differently. There were different selective pressures on both of these things. And so it really does really open up more questions. So yeah, it does lead to mind-blowing reactions.
Well, so it's interesting. You do have a natural evolutionary bent that keeps coming through here.
Yeah.
No, I understand. Mm-hmm. And in this case, so a thought that we were talking about earlier that I'll come back to just to finish it off was that why did the CD4 gene end up having to produce the CD4 class 2 specific co-receptor protein and the CD8 doing the class 1? So maybe there was something special about the CD4 co-receptor protein, and and I don't think so. I think what we came to is that if you reverse the CD4 and the CD8 genes that are making the CD4 and CD8 proteins, you come up with a mouse that does not have a competent immune system.
Right.
Because you can't get the T cells to cooperate with the B cells. You can't clear the virus from the animal either. So I think that what happens is that evolution basically allowed the animals that had made the wrong co-receptor protein in the co-receptor gene loci. When it made the wrong one, these animals didn't have a good immune system and they died.
Mm-hmm. Right.
So the only ones that survived were the ones that were selected by evolution. Not because it needed to be CD4 proteins in the CD4 locus, but because it needed to have a system where the T cells could collaborate with the B cells and the antigen presenting cells could present the virus without getting killed by a cytolytic T cell. That's what I think.
Right.
I just got this. So please let me demonstrate that So I just realized the reason besides the CD4 CTLs killing APCs, they can't kill all the other cells that are virus infected because all the epithelial cells or whatever have
MHC1.
One MHC1, and the CTLs can't recognize it. Yeah.
Yep.
Yeah, you had just figured it out, Vincent, and then everything went black.
Yeah, I was very proud that I figured it out because I this is not second nature to me. So I was looking
Well,
at
it is flip-flopped in our heads. It's all confusing. You really try to wrap your head around it.
Well, I think you need to know it really well to automatically flip-flop. That's the key, right?
Yeah.
Mm-hmm.
I think it's very hard. I think it's very hard to really understand. I think if you go through it very superficially,
Yes.
you think you understand it, but it's so backwards.
Yeah.
It's very hard to understand. I'm really sympathetic to trying.
So, Al, do you expect that many people will request these mice and do all sorts of experiments they've thought of, or do you think not?
I hope so. No, I hope so. I think that people who are interested in understanding, I think people who are interested in TB interactions, people who are interested in antiviral responses, I think they should request these mice.
Definitely.
And when the paper first went online, we immediately we were contacted by people doing TB research in mice, and they were curious to look at at these backwards mice to see how that might affect the TB response. I think there are lots of uses that these mice can have. We still have a number of intellectual questions we want to address. So we've made variations of these mice. But for most of the things that most of the world is interested in, we'd be happy for everybody to get these mice and do whatever they wish with them.
Yeah. So thinking about mechanisms too, these cis-acting elements that are hanging around where this gene is being expressed, are you looking to dive into which ones contribute to the long duration versus the short duration? What are some of your thoughts on that?
My thoughts are I should be. Because these are, these are important regulatory elements. Right. But I think the as an immunologist, I'm really interested in the immunology of these kinds of cells.
Sure.
But yeah, I think if you were interested in regulatory elements, this is a good system.
Yeah, I would definitely think so. Yeah.
Yeah.
So, I'm looking at it from a virus perspective. So the CTLs cannot clear infection. That's because for this virus, CTLs are important for that. But for some, it could be that antibodies are important to clear even, right? You could use these to figure that out. They only make IgM, though, so that would have to be enough, right?
Right. Mm-hmm. That's a problem. These mice don't make good antibodies either, so that's a problem for that part of it.
Yeah.
But who knows with TB? The immune response of TB is complex. Right. No one really understands it. Maybe you need a flip-flop perspective. Who knows?
Oh, I like that. A flip-flop perspective. That's a good title. Yeah. And actually, I don't remember, but did you look at IgA levels? I know we know the IgM and the IgG story, but was IgA also gone?
We did not look.
Oh, see, that's my plug. That's my plug for everyone. Just measure IgA always. Then I don't have to ask you to, and you're going to find out some cool info. So, okay.
Well, yeah, because there's T-independent B cell responses as well.
And actually, I had brought up the whole IgA interacting with the microbiota, but I will say that those are mostly T-independent responses. Right. T cell dependent responses are going to be more your pathogen-specific responses. So I just wonder if you would just still have IgA, T cell independent IgA. Yeah.
But we'll look.
Okay,
that we can do pretty quickly. We'll look, but we've not
and
done
then
it.
Report back. Do you have a Twitter? You could tweet it.
I don't have Twitter.
Okay.
I had a question, and that was, some of the problems in studying the non-traditional cells is that when you mess up the traditional cells, you mess up the non-traditional cells, or they go up or down or whatever. And so I'm just wondering if you can look at more B-independent, T-independent B cell responses where you have normal, you have B cells occupying a niche, right? And you have them around so that you don't have these gaps in the immune system when you've manipulated mice in different ways. Do you envision being able to ask some questions that you wouldn't be able to ask with other mice because they may not be functioning, which actually might be a good thing. So they can't do what they normally do, but they're occupying the space. But now you can look at the functions of cells you couldn't normally address, because if you take away cells, then there's a gap and things change. But if they're there, they're doing their own function, and you also can't look at the unusual cells or the low, abundance cells and their functions with the normal functions there. So are there things that you think about that way?
To answer the question honestly is I don't think about things that way. But but honestly, I mean, seeing you now, I mean, we never really looked at an innate response per se either.
Mm-hmm.
You
Right.
know, and would an innate response be affected?
Yeah. All the innate people listening, they're like wondering what is interferon doing?
I can think about both CD4 and CD8 on some innate cells.
Right.
Yeah.
Yeah. Right.
Yeah. So, I mean, somebody should look for sure.
Yeah.
I don't think it's going to be us, but somebody should look at it.
So I have one more question, and that is that you've been working on this kinetic model for years, and now you finally have like the most definitive data. So is that it? Are you done? You're like the Hail Mary in your career. You're like, I'm done. I've done exactly what I said I was going to do. I've proven my point and pass on the baton.
Well, I'm close, not quite, but close. There's still a number of other things I want to do, and but I'm getting close. Did the strength of signal people have they talked to you yet? Have they called you up? The strength of signal people don't talk to me.
Okay,
it's been odd. But we'll see what happens with the paper. I was happy that the paper got so much attention. Mm-hmm. Not because the paper got so much attention. It was just that this is a discussion that we've needed for in this area for a long time. Absolutely. I actually do think that we've come. I think we're close to understanding how lineage fate is decided during positive selection. But, it's always dangerous to say that. But I do think that we're getting close to that.
So what do you think are the big open questions?
Well, so, I have another in addition to lineage fate, I think the issue of MHC specificity. How does the T-cell receptor get focused on MHC? So I have as many friends in that field as I have in the strength of signal.
Frenemies.
Frenemies, yeah.
So but it's all related actually. It's all these the very first steps that take place during the formation of the receptor, functional receptor, and the first signals that come from that receptor determine the MHC specificity of the T-cell repertoire, determine then the function of all these cells. It's really, everything happens very quickly. So so I'm interested in wrapping that up as well. So we're working on that as well. But so this was lucky because we were able to make a dramatic mouse.
Yeah.
So Chris Hoquist in a comment on in the journal, points out that the mice are stunningly clean. Yeah. Which is to say, they are clean. What's amazing is that all the CD8 cells are helpers and all the CD4 cells are cytolytic. It's quite clean. So you have to be lucky. You have to be lucky to have it turn out that way.
Yeah, it was impressive how much the cells looked like the exact opposite of the data from B6.
Right. If it was 60% or something, you wouldn't have been so impressed. But it was everything was changed. Yeah. Right. So we'll see.
Great. Well, do you have any last things you'd like to say? Any tips or advice for early career scientists?
Well, I think for tips for early career scientists. Well, I think doing science is a tremendous privilege. It's a lot of fun. The day-to-day hassles of getting grants and getting, keeping your mouse colony going and clean is, there are a lot of hassles to it, but it's an awful lot of fun. I think that the most important thing I would say to young scientists is don't believe everything that other people tell you. Think for yourself. Think for yourself, and your ideas are as good as anyone's. So that I would and disagree with the experts. There's no great glory in proving an expert correct. Prove them wrong, because they are wrong. So that's what I would say.
So it's good advice for late career people too.
Yeah, I think it's true. I think that is true.
Al Singer from the National Institutes of Health, thank you so much for joining us.
Well, thank you very much for having me. I enjoyed this. I'm sorry to have chewed off your ear on this.
Oh,
not at all.
Could go
That's what we wanted.
another hour. Yeah, we loved it.
Okay, good. Thank you very much.
Another episode where I'm going to have to rewrite part of my immunology lecture.
That's right.
Yep.
Cindy, come down and buy me a coffee.
I will.
And you can come to the lab and do a few experiments.
Oh,
Yep.
there you
I'll
go.
have to do some TLR signaling in the flip-flop mice.
Okay, good idea. Okay. All right, guys. Thank you.
Bye-bye.
See you.
Bye.
Bye.
So you said people with IgA deficiencies are pretty much okay, right? And that's what I've heard for SARS-CoV-2, right? They don't do any more badly than. So what's this emphasis on immunity then? What the hell? Tell me.
Well, here's what I think we can say is that you're not going to die from a viral infection if you don't have IgA. Right,
Okay.
because these individuals are not dying. But I don't think we understand fully the nuance of, infectious titers, transmission. We don't fully understand what is happening with IgA in the mucosal space. And I think it's because those individuals haven't been studied at that depth. Sure, we can look and say, okay, they're not dying in greater amounts. But what is the proportion of those people? How old are they? There's a lot of that nuance. So yes, that would be what I would say to that.
So, Steph, one thing that I've heard about this, and you can tell me if I'm totally incorrect.
Oh, you're probably right.
Is that evolutionarily, there may not have been a lot of selection against it because of people having IgA from mom with breastfeeding, at least very early.
That's very interesting. So I could see that, but then also because there's IgM and because IgM can also show up in the same places that IgA can. But I go back to this thing, we talk about, there's a lot of dogmas about IgA. Oh, it's not durable. Oh, maybe it doesn't matter. But I don't, just like what Al said, when I ask people, I go to these like flu civics meetings, I go to these SARS, I ask, have you measured IgA? No one's measuring it. No one's looking at it. So I think we need to study it more. Yeah. Now the microbiota people would definitely say it matters because I think that people, especially babies, can have a lot of disturbances in their gut leading to things like necrotizing enterocolitis. And so it goes back to Brienne's point about, the evolutionary aspect of maternal IgA.
But if T cells can clear an infection, if you don't have IgA, you'll get infected. The T cells and the other antibodies can help. So if you want to prevent infections, you may need IgA, but most of the time we don't do that. So
here's a scenario where I could see IgA being effective. So let's say you have Omicron, right, which has mutated 30 times and it's evading our antibody responses. If IgA can bring down the viral load in your mucosal compartments even 2 logs lower, that's less amount of virus that could seed into the lung. So I think it is at this kind of reducing the viral load, reducing transmission where it matters.
Yeah, but that usually doesn't happen in a post-infection, post-vaccine scenario, right? Our vaccines don't do well with IgA, but we're still not dying and we're not going to the hospital. So I agree. I understand what you're saying, but I don't know how you achieve that, right? Maybe you could make a vaccine that would give you durable IgA. I don't know. Right.
Well, yes. Go ahead, Brian.
I also think that I totally see what you're saying, Vincent, but I would actually be interested to know do we have data on the vaccine responses in those IgA deficient individuals with Omicron?
Yeah.
While we can say, oh, I can't measure a massive role for IgA, if we look in those individuals and we were to study them at really great levels of detail, would we find something that's different that maybe we didn't expect IgA to have been doing?
Right.
Yeah, I think that's worth it.
And there's some interesting data that I'm on some call, and they have a bunch of clinical data that individuals that were protected, serum levels did not correlate, but IgA levels did correlate. Again, it's just because it hasn't been measured before. So like what's going on there? But you're right. The selective pressure of dying probably isn't going to select IgA because IgG can just be in the lung and be there and protect you.
Or T cells, right?
Or T cells. But I do think there's this, thinking of the concept of like I think IgA can impact the evolution of virus transmission. And that needs to be studied.
Because it could bind it on the way out just as easily as it could bind it on the way in.
And exactly. And if you have IgA that can be bound up a viral particle that's transmitting from me to you, and you don't get infected because it can't bind to you, that's a pressure. So, yeah.
I agree. I think it should be studied. You should do that, Steph.
I know.
Have a long career studying IgA. However, one of the best mucosal immunogens is poliovirus, and it doesn't prevent shedding. It sheds like crazy, but you're fine. It doesn't matter,
Right.
So,
right.
Yeah.
Polio.
However, norovirus, respiratory syncytial, those all would benefit from IgA, mucosal IgA, but and so it depends on the virus or the pathogen that you're going to look at.
Yeah.
Too.
So I sometimes think about antiviral immune responses as you need two arms, right? You need to prevent new infections, and you have to clear the infections that are existing.
Yeah.
And so they need to work together, because if you only have one, it's going to be really hard. So if you only have antibodies, you're primarily going to block new infections, although you have antibody-dependent cell cytotoxicity, right? But if you only have T cells, you'll kill virally infected cells, but you're not going to prevent new viral infections. So you're going to cause a lot more immune damage. So the antibodies and the T cells work in concert. That's how I think about it.
Yeah.
Yeah.
However, with Omicron, and if you've had 2 doses of vaccine and the antibodies are not very good, you still survive. Not everybody dies because of the T cell response. So it really depends, I think,
And then there's the other thought of, okay, we talk a lot about death, but if you can reduce the viral load in your upper respiratory tract, can you reduce long COVID? Can you reduce post-inflammatory reactions?
Yeah. And there are things like, the memory B cells as well that are probably doing something, not just the antibody level that's present in serum.
And I think why intranasal and IgA is getting a lot of attention. 1, it's understudied, but 2, never before have we really put such an emphasis on PCR positivity and this transmission.
Vaccines don't prevent PCR positivity, right?
Correct.
But I think I agree. I think the attention on IgA is great, but I think a lot of people have the wrong reason for paying attention.
Well, I think that might be because, some of the initial studies that came out when they first had the vaccine, they did show significant reduction in PCR positivity, because they were looking at like hospital people going to the hospital who are being surveillance tested. And those that had a vaccine were much less likely to test positive than those that didn't, right? So that was the data that we were hearing early on, because it was perfectly matched in the first wave, right? But now people have that in their mind that's the way it should work. When
but that was also within 6 months of the first vaccines, and when you had high antibodies, right? So it's a mess.
Yeah, interesting.
Right.
Interesting. Okay, well that was fun. Yeah,
thank you. And let's so microbe.tv/immune is where you can find the show notes. You can send questions and comments to immune@microbe.tv. And if you like what we do, consider supporting us. Microbe.tv/contribute. Your contributions are US federal tax deductible. Cindy Leifer's at Cornell University. Cindy Leifer on Twitter. Thanks, Cindy, for getting Al today.
Of course. Yeah, that was really fun.
Steph Langle's at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Thank you. This is so fun.
And Brianne Barker's at Drew University. Bio Prof Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.